Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I'm your biology educator. And yes, as you can see back on the back of my screen, yes, very important information guys. On 30th of August, we are starting with the first CBT. Formally, the first CBT is going to be conducted on 30th of August and you have to get yourself registered for that if in case you have not done that yet. So yes, if you are already an iconic pro, then you have already taken the subscription. You have to pay nothing. Just make sure that you get all the formalities done exactly in the same format as you are going to do for NEET 2027, which will be in the CBT format. And no doubts about it. Still, some of you are asking, ma'am, whether NEET 2027 is confirmed to be in CBT. Yes, it is confirmed. It was long confirmed before NEET 2026 as well. Right? So yes, it is CBT and you have to get ready for it. Iconic Pro students are all set already. They have already taken the subscription, and those of you who have not taken it yet, guys, if you are a plus student, get it upgraded by paying 4,999 extra. And if you are an iconic student, then you have to pay 2,999 for the first CBT exam in your nearest offline centers. Attempt offline CBT at 7. There'll be 70 plus centers all across India. 13 offline and 15 will be online. So you have to get into that zone. You have to get into that practice, how to download the admit card, how to go there, get all the things done and be ready for NEET exam. All right, so make sure that you do that at the earliest if it is not done yet. Good evening all of you. Yes, I am back, and guys, we have already started with the backlog series. In the part 1 we have studied about cell, the basic structural unit, right? The historical background, we have seen cell theory, we have studied it all. Then size difference, shape difference, overview of the cell I have already given you, and we have already started with the we have seen the difference between prokaryote and eukaryote, and we have started with the details of prokaryotic cell. After completing the cell envelope, I have started discussing the cytoplasm, and see, this was all about this plasma membrane modifications, and then fimbriae, then flagella, all done, and we were discussing cytoplasm. Please let me know that you have done it till here with me and everybody is ready for the next part. Yes guys, good evening all of you. Quickly share this session with all your friends, and we can start ahead. Done? Now, so cytoplasm guys, in cytoplasm we know, in cytoplasm there are two main components. One is ribosomes, and second structure which is present in cytoplasm is the inclusion bodies. It is the inclusion bodies. Now, inclusion bodies are meant for storage purpose. What is the purpose for inclusion bodies? They are meant for storage. Is that clear? They are meant for storage, and just like this one is also not membrane bound, so membraneless. These are also membraneless. So both the structures present in cytoplasm of prokaryote is membraneless, right? And they lie freely in the cytoplasm. Lie freely in the cytoplasm. Noted? And then what all structures are included in inclusion bodies? What all do we consider under inclusion bodies? So it includes number 1, we have phosphate granules. So they are storage and they will secrete also. So phosphate granules. Number 2, Synophycean granules, glycogen granules, and gas vacuoles. Now, very important part is that you have to remember which all bacteria has these gas vacuoles. Number 1, I'm writing it here. Number 1 is blue-green algae. Blue-green. Then purple bacteria and green bacteria. So these bacteria, these prokaryotes have gas vacuoles. So these should be well learnt. Noted, everyone? Good evening, all of you. Good evening. Yes, guys, I've just started. We have already completed till ribosomes. We are starting fresh from inclusion bodies. All clear? Now, moving on to the next part. So, cytoplasm has this much only. Cytoplasm has this part only. Now, see, in the previous session, I have already told you about the genetic material. Yes. So, we have talked enough about cell envelope. We have talked in detail about the cytoplasm. There isn't much compartmentalization, no compartmentalization at all. And there is not much in this. Ribosomes, inclusion bodies, all done. Now coming on to the genetic material, already told you it does not have a well-defined nucleus. And the genetic material lies freely in the cytoplasm. And that region is known as nucleoid. And then there is an extra chromosomal genetic material known as nucleoid. Right? So I'll show you diagrammatically how does the genetic material of prokaryotes look like. Genetic material in prokaryotes, right? Now, let's say this is the bacteria, right? And this one is the genetic material. This is the genetic material number 1, and this is the genetic material number 2. So the first thing is, this is called nucleoid. So genetic material is present in two forms. One is nucleoid and second is plasmid. Noted? The main genetic material is this. The main genetic material is nucleoid, right? Now what is nucleoid basically? So as far as you see that genetic material is extended, this whole region is known as nucleoid. What is it made up of? It contains double-stranded DNA. But do you see that I have started from here and I have brought it back to this point? So there are no free ends. It is circular. It is of very small amount. It is in very small amount. Is that clear? And of course it is DNA, and DNA is acidic in nature. It will not feel stable without the presence of some basic proteins, right? So it is associated with, this DNA is associated with basic proteins, and these are not histones like in eukaryotes. Basic proteins known as amino proteins, and these amino proteins are in the form of hooks. They are in the form of hooks like this. Let's say these are amino proteins in the form of hooks on which this DNA is suspended, and that's how these amino proteins stabilize the DNA. Is this point clear? Now because it is of very small amount, it is in very small amount, so we need some extra chromosomal genetic material also, which is nothing but plasmid, Okay, now plasmid is also DNA, but other than this, so I would call it extra chromosomal genetic material, right? This is extra chromosomal genetic material. Extra in biology does not mean extra, it means outer side, outside the chromosome. By the way guys, we will not actually use the term chromosome here, we call it prochromosome. Chromosome term can only be used when histones participate. So prokaryotes cannot be called, the DNA of this cannot be called as chromosome. We would rather call it pro-chromosome, means primitive form of chromosome. Prochromosome, you can call it genophore, right? All these are terms for this. Noted, right? Now coming back to plasmid, this is the extra chromosomal genetic material, and it also contains very important genes, By the way, similarity to nucleoid is this one also contains double-stranded circular DNA. So that part is common. double-stranded circular DNA. DNA. Is that clear to all? Please tell me these points are clear to everyone. Yes, please. Points clear to all? Now, because as I said, it contains very important genes. Now, these genes could be related to fertility, or fertility, then we'll call it F factor. So, F factor is a gene related to the fertility. Okay, then it may have antibiotic resistance genes, and many more. Is that clear? So they are found in this plasmid, and this plasmid will be of huge importance when we talk about biotechnology. It can be used as a vector. It is, in fact, our biotechnology will not be even halfway there without the use of this plasmid. Hashtag important vector in biotechnology. Vector means it is going to carry genes. How, why, where, we'll talk about it all. Is this point clear to all? Right? So this is about the genetic material. Quickly take a screenshot, note down, and let me know that it is done. Yes, guys, quickly let me know this is done. So with this guys, we are done with prokaryotic cell. We are done with the prokaryotic cell. Noted? Now starting with the eukaryotic cell. Yes, starting with eukaryotic cell. See, we know that eukaryotic cell will also have the basic same composition, that is it will have cell boundary. Now the cell boundary of eukaryotic cell will be made up of not glycocalyx but only cell wall and plasma membrane. Right? Then another component is cytoplasm, which consists of course the cytosol, the jelly like material, right? Then organelles, and other than organelles it will have some cytoskeletal structures. So these are the three components of cytoplasm. So as we know, cytosol is jelly-like, right? This is a common feature, semi-fluid in nature. So cytoplasm is always semi-fluid, be it prokaryotes or eukaryotes. Noted? Then as far as organelles are concerned, guys, now we can divide these organelles on the basis of the membranes. You will see that there are some organelles which are, double membrane bound, while there are others which have single membrane, and also there are few which do not have membrane at all, so they are membraneless. So that's the categorization of organelles, right? So in double membrane we just have two, and I know we all know about these two, mitochondria and chloroplast. While membraneless, in membraneless, guys, we have of course ribosomes and we have centrioles. Okay, and as far as single membrane bound organelles are concerned, the single membrane bound organelles, by the way, these are ER, Golgi body, lysosomes and vacuoles. So these four come under this category. Is that clear? And last not the least, cytoskeletal structures means those structures which are going to form a skeletal work. So we have three components in here. 1, 2, 3 microfilaments, microtubules and intermediate filaments. So we are going to discuss each one in detail from cell wall till the end, intermediate filaments. Noted. And last not the least, the third component of cell is the genetic material, and we know that the name eukaryote has been derived from the true nucleus. So basically there is a true nucleus. So genetic material will be found mainly in the true nucleus, which is membrane bound, right? So there will be true nucleus, but apart from this you will also see some genetic material in structures like mitochondria and chloroplast, right? So what is the significance of genetic material there, and why is it there, how it became a part of these two structures? We'll discuss it all. Yes guys, quickly let me know that these points are clear to everyone. Anyone with any doubt? Noted. Now the clear-cut difference between the genetic material that I have just discussed in case of prokaryotes and now in eukaryotes, the difference is here it will be, you know, membrane bound. The genetic material is membrane bound. And this membrane is going to be double membrane again. Then while teaching you about the genetic material of prokaryotes, I told you we cannot call it prochromosome, right? While here we can call it chromosome. So they are condensed into the form of, they are condensed as chromosomes. How, when, why, where, we'll discuss it all. By the way, this genetic material is also in the form of double-stranded DNA, but now, unlike prokaryotes, this one is not circular, this one is linear. There was only single prochromosome there. Here there are multiple. They are multiple in number. For example, we as humans, we have 46 such structures, 46 chromosomes, right? Then multiple in number, double-stranded, linear. And in order to stabilize this double-stranded DNA, this is stabilized by histone proteins. So they are associated with histone proteins, which are basic in nature to stabilize the acidic DNA. Are these points clear? Please tell me if there is any doubt. If there is any doubt, please let me know. Yes, guys. All clear? Noted? Right? Can we start in detail now? All right. Starting with cell wall. I'll start with cell wall. So cell wall, in case of prokaryotes, you should be able to recall, guys, cell wall in case of prokaryotes was made up of peptidoglycan, right? Of course, a carbohydrate, but sugar plus amino acids, right? But in case of this eukaryotes, now cell wall will be present, and different type of eukaryotes will have different composition of cell wall. First of all, the similarity between the cell wall of prokaryotes and eukaryotes is both are non-living, both are rigid, that means non-flexible, both are freely permeable, right? And what is the function of cell wall here? The function of cell wall is, there are 4 functions given in NCERT. So the 4 functions, write down. Number 1 is it gives shape. It gives shape, right? Number 2, it protects from the mechanical damage. Protects from mechanical damage. Number 3, it's going to help in cell-to-cell interaction, and also it will act as a barrier for large molecules. Barrier for large molecules. See, although we have said it is freely permeable, so it will not take care of the aspect whether which molecule is entering or taking an exit. So in general it is freely permeable, but it becomes a barrier for large macromolecules. Please tell me these points are clear to everyone. So this is about cell wall. Noted? Now Cell wall is found, where is it found? It is found in many types of cells. For example, it is found in, number one, you will see it in fungi. And in fungi, it is made up of a sugar known as chitin, right? It is found in plants. In plants, it is made up of cellulose, the main component, but other than cellulose, it has hemicellulose and pectins, right? In some protists also you will see it made up of silica like in diatoms or cellulose like in case of dinoflagellates, right? So cell wall is present in these. Then animals will never have a cell wall, of course. Then algae. In algae, the cell wall is made up of galactans, mannans, cellulose, and very important, which you tend to forget, calcium carbonate. Is this point clear? Rather, plants will have pectate salts, calcium pectate, magnesium pectate. We'll have pectins in plants, but calcium carbonate will be present in the cell wall of algae. So this is where it is found. It is found in. Is that clear? So different cells will have different components, components in their cell wall. Noted? Anyone with any doubt? Right? Now I want to tell you that the cell wall which is present in the young cell is called the primary cell wall. Now see here. So young cell, see, young cell has primary cell wall, right? But later on, this primary cell wall diminishes and instead there is a secondary cell wall develops. A secondary cell wall develops. This is about mature cell. In mature cell, primary cell wall will diminish and the secondary will develop. Now, where is it exactly going to develop? And let's see that through a diagram. Now, let's say this is one cell, right? This is another cell. And this one, this green thing, this represents the plasma membrane. Noted? Now, outer to this plasma membrane, there is present, outer to this plasma membrane is present the primary cell wall. So this is the primary cell wall. By the way, guys, there is no gap in between plasma membrane and cell wall. I am just drawing it separately, far apart to distinguish it properly. Is that clear? Otherwise, they are intact. No doubt. So this is the primary cell wall. Noted? Now, as I said, as the plant grows older, this one will diminish, rather a secondary cell wall will develop. Now note down, where will secondary cell wall develop? Secondary cell wall will develop between the primary and the plasma membrane. So that's where the secondary cell wall will develop. Very important line that you have to remember, which NCERT says that it is present, the secondary cell wall. Note down, the secondary cell wall is present inner towards the membrane side, of cell. This is NCERT line. So you must know how to decode it. See, inner towards the membrane side. Membrane side means towards the plasma membrane. Inner to primary cell wall and towards the plasma membrane. Are you able to decode this? This is NCERT line, right? In short, if I have to say, then I would say that secondary cell wall, the secondary cell wall is between, it is present between where? Plasma membrane and primary cell wall. But eventually this primary cell wall is going to diminish. Is that clear? Now, next point is that how are these two cells connected? So guys, there is this wall which connects the two cells. So this wall which connects the two cells, this one is known as middle lamella. This one is middle lamella. Lamella means membrane. So it is present in the middle of two cells. So remember that middle lamella is common to two cells, common between two plant cells. Is this clear? And now if you want to connect the two cytoplasm, then see here, then you have this connection, which is known as, this bridge is known as plasmodesmata. So that's how the components from A cell can move into B cell. Are these points clear? So this bridge, this intercellular connection is known as plasmodesmata. Please tell me these points are clear to everyone. Yes, guys. These points are clear to everyone, right? So when I was telling you that cell wall in case of plants have pectin salts, so it is present in here. So this middle lamella, what does it do? It is common and it glues the two cells together. And it contains calcium pectate or magnesium pectate salts. Are you getting that? Please tell me these points are clear to everyone, and with this the cell wall is taken care of. Yes, guys, are these points clear? Now I want everyone to spare 1 second, those who are just sitting back quietly and writing down the answers and writing down all these notes and not participating in the chat. Well, I do appreciate to some extent, but now spare 1 second and let me know in the chat. Yes, guys, I need thumbs up from everyone. Are these points clear to all? Quickly let me know. Are these points clear to all? And please don't hesitate in leaving your good comments and letting us know how much these lectures are helping you. And also, guys, on the first coming up CBT, that is on 30th of August, this chapter is a part of CBT, cell cycle, right? All these topics are coming in your first CBT. So reminding you once again, you have to get yourself registered for it. And if you're already a student of Iconic or Plus, then you have to get it upgraded. Is that clear? So 30th August, guys, you have your first CBT. So get ready for that. Download your admit cards exactly in the same way as you did for your NEET exam, re-NEET exam, or you're going to do it for your coming up CBT 2027, right? So get used to it. Things that we get used to, they stop scaring us. So instead of working under that tremendous pressure, let it be part of your whole life now, because you're becoming doctors. If you yourself will stay in that anxiety attack, how will you treat your patient? So get normalized. Now, guys, with that, moving on to the next structure, which is plasma membrane. You can call it plasma membrane, plasma lemma, or cell membrane, call it anything. So plasma membrane, by the way, is a living membrane. Okay, this is living membrane, this is flexible. Why it is flexible, how it is flexible, we'll talk about it. Flexible, and it is semi-permeable. These are all the features opposite to that of cell wall, right? Cell wall was non-living, it was rigid. Cell wall was freely permeable. Now you see the differences. Keep noting down the differences. Is that clear? Done. Now, plasma membrane, the detailed structure was only studied after the discovery of electron microscope. Detailed structure studied after electron microscope discovery of electron microscope in 1950s, and that's when many scientists tried explaining the detailed structure of plasma membrane. But the one which is most accepted model of plasma membrane is Singer and Nicholson's model. Singer and Nicholson's model, known as fluid mosaic model. Now, this fluid mosaic model was given by Singer and Nicholson. Singer and Nicholson in 1972. Okay. So these are the basic things. Now, according to this fluid mosaic model, why do we call it fluid mosaic model? We'll come back to this. But according to this model, first let's see what is your plasma membrane made up of. So plasma membrane is made up of three components. Number one, mainly it is made up of proteins, then lipids, and third is carbohydrates. Lipids are present in two layers, so we'll call it bilipid layer or bilayer, right? So first of all, let me draw this lipid structure. So this is one layer, this is one layer of lipids. Okay, this is lipids, and then there is another layer, this is also lipids. So there are two layers of lipids. Noted? And it goes on. I'm going to save time and not draw it all. So this is lipid. Okay. Now remember, the most common type of lipids which form the plasma membrane, they belong to the category of phospholipids. These are mainly phospholipids, means lipids associated with phosphate group. Phospholipids, noted. Second most abundant is cholesterol. So main it is phospholipids and second is cholesterol. Okay, so these lipids that I'm drawing here, they are majorly phospholipids. Noted. Now listen, you can see here the lipids I'm drawing. Let me draw it here also. So this one, note down, this one is the head and this one is the tail, right? Similarly, this is the tail and this one will be the head, right? Now see, on the outer side, towards the outer side of the cell, there is fluid, interstitial fluid, and even in the cytoplasm there is fluid. This is aqueous medium, water-based medium. So it means head cannot be water-hating. Head is hydrophilic. Head is hydrophilic. It is water-loving. Is that clear? Head is hydrophilic. Please tell me that these points are clear. And it is polar in nature. While the tail, it is away from water, so tail is hydrophobic. Tail is hydrophobic in nature. Please tell me that these points are clear to all of you. And it is made up of non-polar saturated lipids. Yes guys, so lipids, which these phospholipids, they are saturated in nature and they're present in this way. Are these points clear to all? This is all about lipids. Noted, which are the two types of lipids that I've already told you. Main one is phospholipid, the second one is cholesterol. So cholesterol will also be present somewhere here like this. So these are cholesterol molecules offering stability. Is it clear? This is cholesterol. Now, the second component present in the plasma membrane is proteins. Now, proteins, guys, see here, proteins proteins can be differentiated on the basis of their ease of extraction. That means, I'll tell you, the proteins which are present on the outer side like this or towards the inner side into the cytoplasm, that will be this, right? So these are easily extractable, right? So these ones are known as peripheral proteins. These ones are known as peripheral, on the boundary, or you can call them external or extrinsic proteins. Is that clear? This one as well as this one. While there are few proteins which are, present within the membrane like this, or in fact they can be present like this entirely from between the two layers, trans membrane proteins, right? So these are different types of proteins. Noted? So the proteins I have drawn in blue, these ones, these are called integral proteins. Integral because they are integrated with the membrane, within the membrane, sorry. So these are integral proteins. Now another point about integral is that these integral proteins could be completely integrated or partially integrated, right? These could be completely integrated or partially integrated. Of course, you can see the difference, that this one is partially integrated while this is completely integrated. These completely integrated ones can be known as tunnel proteins also. Please tell me these points are clear to you all. So this is the basic structure of plasma membrane, and it is common for both prokaryotes and eukaryotes. Is that clear? And then the third component, which is carbohydrates, right? So this is in the form of oligosaccharides, which are associated. Oligosaccharides means small, not monosaccharides like glucose and fructose, but such sugars of 2 to 9 molecules in number, they will combine to form a chain, right? Oligosaccharides, and they will be associated with, they are associated with oligosaccharides. They are associated with both proteins and lipids. With both proteins and lipids like this. So they can be present here as well as like this. Are you getting that? So these are oligosaccharides. Now, because every cell is different, so obviously the ratio of this proteins and lipids is going to be different. Not every cell will have a higher protein content as compared to the lipid content, or the vice versa. It will vary from each cell. Is that clear? It will vary from cell to cell. But in case we are talking about human RBC, in case of human red blood cell, the ratio of proteins to lipids is 52:40, or you can say proteins 52%, lipids 40%. Please tell me these points are clear to you or not. So this ratio is going to differ. The ratio will differ like this. Please tell me, anyone with any doubt, quickly let me know in the chat, guys, and we can proceed. Quickly let me know in the chat and we'll proceed. Done? Clear to all? Noted? I will ask questions, and in fact CBT will ask questions on these topics. First, let's complete the chapter, then I'll ask you questions. And guys, we'll be practicing the questions in Unacademy Learners app, free of cost, special classes. So don't forget to join me there, right? And test practice is super important. Just pen and paper mode will not help this year. I'm telling you very frankly. Is that clear to all? Noted? And once again, those of you who have joined me late, guys, first test. Get ready for the CBT on 30th of August. And those who have not upgraded yet, please get it upgraded and make sure that you use the code TR Live. If you have not taken the subscription, you have to, and then upgrading it will also require the code. Use the code. Noted? Now, talking more about plasma membrane. Now, let's understand why did we name it as fluid mosaic model. Why? See, first of all, mosaic word means mixture. Mosaic means mixture. So it is a mixture of proteins and lipids, right? It is a mixture of proteins and lipids, and of course carbohydrates too. But these are the two main components. Why do we call it fluid? So what does fluidity means? What does fluidity means? In general, fluid is something that flows, right? Now what is fluidity with respect to the plasma membrane? See that. So fluid nature. So membrane is not like completely fluid. It is quasi-fluid in nature. Quasi-fluid means semi-fluid. It is quasi-fluid in nature. And it's very, very important that this quasi-fluid nature hashtag allows lateral movements. This is NCERT line. Lateral movement of proteins within the bilayer. What does this mean? Within the bilayer, this is a direct question from NCERT. So what is the quasi-fluid nature? The fluidity of plasma membrane allows lateral movement. So fluid means what can flow. Now what is the real meaning? The real meaning is that the location of these proteins is not fixed. This protein can move here, this protein can move here, this protein can move here. So the proteins can move like a fluid within this bilipid layer. And also I must tell you that even the location of this each lipid molecule is also not fixed. They can also flip-flop. This one can move into the upper layer, upper one can go here, this one will move here, this one will move, take its place. So everything is mobile, and it is because of this fluidity. Note down. Now what is the use of this fluidity, guys? The advantage, the importance of this fluidity is, number one, it allows, see, the fluid nature, I'll write it here only, the fluid nature allows, number one, endocytosis. Endocytosis means absorption of substances into the cytoplasm. Then cell growth. Third, it will allow secretion. Fourth, it allows cell division. And fifth, it allows intercellular junctions, formation of intercellular junctions. All these are possible because your membrane is not fixed, it is mobile. Yes guys, please tell me these points are clear to everyone. Noted? Is that clear to all? Yes, please. Done? So yes, this was about plasma membrane. Now, as I said, your membrane is semi-permeable. It is semi-permeable. So it means not everything can just move in and out without any regulation. So how do we regulate certain things? So what is the method of transport? So methods of transport across the plasma membrane. Transport across plasma membrane. Now see here. So first of all, guys, you can divide the methods into two categories. One is active transport and second is passive transport. I am sure that we do have this basic understanding that active transport is the movement of substances from low concentration to high concentration. So that requires ATP, right? While in case of passive, the movement is from high to low. So there is no ATP required. Done? Now, active transport, listen, active transport occurs for some ions. For example, ions like sodium, potassium, and they move with the help of ATP, using ATP, right? So the example is sodium potassium pump, right? Sodium potassium pump. Whenever you come across this term, na, pump is always energy utilizing structure. Okay, so it is sodium potassium pump, active transport. Now, as far as this passive transport is concerned, we have three categories in here. We have three different ways. Number one is diffusion. Number two is osmosis. And number three is facilitated diffusion. Facilitated diffusion, right? Diffusion, osmosis, and facilitated diffusion. Now, diffusion is for neutral solutes. For neutral solutes, we have this simple diffusion, neutral solutes, right? Then osmosis is shown by water, while facilitated diffusion means, of course, it will move from high to low, but it will require a carrier protein. So this is in the presence of carrier protein. That is why facilitated, we use the term facilitated, means using the facility of carrier protein. And the example here is polar molecules. Now you will get direct question also based on this, direct question. Please tell me these points are clear. And guys, with this, the structure of plasma membrane is also done. Yes, guys, is this clear? Yes, Haripriya, Nancy, Kirtana. Guys, stop chit-chatting and please let me know. Are these points clear? Everybody is done with it? Noted? And points are clear, differences are clear. I'm telling you the type of question side by side. Yes? I hope you're making the notes also along with me. Cool? All right, guys. So this was about the cell boundary. Now moving on to, first I would like to discuss the genetic material. Or let's say nucleus, and then we'll talk more about the cytoplasm. So we've talked about the boundary. Let's talk about the brain of the cell, which is nucleus. Now we know that the first nucleus was discovered. It was first discovered by Robert Brown. This was first discovered by Robert Brown, right? In 1831. Can anyone tell me where exactly, in which cell did he discover the nucleus? Anyone? Please let me know in the chat or in the comment section. Can anyone tell me in which cell Robert Brown discovered the nucleus? Yes, guys. Now, in order to study nucleus, he had to stain it, stained with basic dyes. It has to be, now if we want to study nucleus, we have to stain it with basic dyes, and therefore it was known as chromatin, and it is known as chromatin by Fleming. Yes, guys. So the name chromatin was given by Fleming. Noted? Also, the best stage to study the structure of nucleus, is, yes guys, who can tell me that? The best stage to study nucleus is interphase. That means when the cell is not dividing yet, interphase is the non-dividing phase. Because once the cell enters into the dividing phase, the whole overall entity of nucleus will be dissolved. Nuclear membrane will dissolve, right? The whole genetic material will come into the cytoplasm. So if you want to overall see the structure of proper structure of nucleus, how it is when the cell is not dividing. And also another fact is that most of the time, 95% of the total time of a cell is in the non-dividing stage, interphase. So how is nucleus in that non-dividing stage? So therefore the phase is known as interphase. Is that clear? This is the best stage to study the nucleus. Anyone with any doubt? Clear to all? Now, next point is, what are the components? So there are 5 components which in total make nucleus. 5 components. Number 1, nuclear membrane. Number 2, nuclear matrix. That is called nucleoplasm. The third is the genetic material, which is in the form of threads, chromatin threads. So yeah, chromatin threads only. Then there are pores which help in the transport of things in and out of the nucleus. So we call it nuclear pores. And very interesting structures called nucleolus. These 5 components can be seen within a nucleus. Please tell me these points are clear. Anyone with any doubt? Yes guys, quickly let me know. These are the 5 components. This is the best stage to study the nucleus, and this is how it was discovered. You have to tell me in the chat or in the comments, guys, in which cell nucleus was first discovered. You have to tell me that. Noted? Now I am going to draw the structure for you to understand this. So let's say this is nucleus and the nuclear membrane. So this one is the nuclear membrane. This is the nuclear membrane, right? As you can see, it is a double membrane. So double nuclear membrane. Do you see the gaps in between? These structures are called nuclear pores, right? Then this matrix is known as the nuclear matrix itself, or you can call it nucleoplasm, right? And then there are some structures present within the nucleoplasm. These are called nucleoli. Please tell me these points. These structures are clear, the labelings are clear to all, and then I'll start with the details. I have not drawn the chromatin fibers yet, but don't worry, we'll take care of it in the next slide. Yes guys, are these points clear? Now what about the nuclear membrane? Already told you it is double in nature. It is double. Now the two membranes run parallel to each other, are parallel to each other. Is that clear? And do you see that there is a space in between? So this space is known as space between the two membranes, is known as perinuclear space. We'll call it perinuclear space. And what is the distance, like what is the value? It ranges between 10 to 50 nanometer. Anyone with any doubt, please tell me. Noted? Then guys, there are some ribosomes also attached to the nuclear membrane. These are ribosomes. You will see ribosomes attached to the nuclear membrane. Clear to all? Right? Now, Do you see when the two membranes are fusing with each other, it leads to the formation of nuclear pores. So it is formed by the fusion of two membranes, right? And why is it important? It will help in entry and exit. So intake and exit. So helps in transport, helps in movement of RNA and protein molecules and in both the direction as per NCERT. So you have to remember the NCERT word by word. Is that point clear? Yes, guys. Very good, Pradeep. Pradeep has answered me correctly, but okay, let everybody answer. What about a nucleoplasm? So nucleoplasm is nothing but the nuclear matrix, that means the ground substance where all the nuclear activities occur, right? And it is this nucleoplasm which contains the genetic material as well as the nucleoli. Please tell me that these points are clear to everyone. Yes, guys. Are these points clear? Done? All right. Last not the least, on this slide we have nucleoli. Do you see these are two or more spherical bodies? They don't have membrane. These are also membraneless. Why? Because it is basically rRNA folded. It is made up of RNA and helps in, it will help in the formation of RNA. Hashtag involved in active rRNA synthesis. Active means a lot of rRNA synthesis is occurring here. So rRNA synthesis means hashtag it is a factory of ribosomes. See, ribosome produces proteins, but proteins producers, that means ribosomes are also themselves produced somewhere. So where are the components of ribosomes formed? In the nucleoli. Is that clear? And this is also membraneless. Please tell me these points are clear. So the number of nucleoli, you'll usually see one, okay, but the number may be more than 1, 2 or 3, not a lot, like 2 or 3, then the number will show you that whether this cell is involved in protein synthesis or not. So more the protein synthesis, more will be the number of these nuclei also. More the protein synthesis, more is the ribosomal component needed, more will be the nucleoli. Are you understanding the link? Please tell me, guys, is this point clear? And then in this nucleoplasm, guys, we have these chromatin fibers. We have this material, genetic material, in the form of chromatin fibers, right? Let's talk more about chromatin. Yes, please. So talking about the genetic material of the chromatin, already you know that it is double-stranded. There are histones associated, right? Let's talk little more about it in detail. So chromatin, it is made up of, so it contains, or it is made up of, number 1, DNA, histones. Other than histones, then there are non-histone proteins also needed. Non-histone proteins. These are also proteins. By the way, histone proteins are only unique to eukaryotes, right? And then 4th component is RNA. So there are these 4 components which chromatin is made up of, right? Then this chromatin is, see, as long as it is just loose, so this is loose nucleoprotein fibers. These are loose nucleoprotein fibers, but as it moves towards, as the cell moves towards division, right, this chromatin is going to condense. It is going to condense to form chromosomes. It is going to condense to form chromosomes. Is this point clear? It is going to condense to form chromosomes, right? But this happens only during the cell division. Is that point clear? Right? Now you must be thinking that nucleus is the brain of the cell. Yes or no? But it is the brain of the cell, because all the regulations are going to occur here, brain of the cell, because it contains DNA. DNA will change into mRNA, mRNA into protein. So DNA will express itself in the form of proteins, right? Now, brain of the cell, fine. But sometimes in few cells, this brain is absent, right? There are few cells which don't have nucleus. Why? See, now that is basically an adaptive measure. Initially, the cells do have nucleus, or their precursors have nucleus, but eventually they tend to lose that nucleus during differentiation. Now, this is a part of their differentiation. This is a part of their specialization journey, right? For example, few enucleated means in which nucleus is absent. Few enucleated cells are number one, we have sieve tube cells in angiosperms. See, it's a part of phloem. They don't have nucleus. That is why they are accompanied by companion cells. And second, your mature RBCs don't have nucleus. So mostly RBCs of mammals don't have, mostly, not a rule. Are these points clear? Anyone with any doubt, quickly let me know. Quickly let me know. Is this point clear? So these are the enucleated cells. While similarly, there are few cells which have more than one brain. Multinucleated condition. Can you name that? Any multinucleated cell that you know of? Please let me know in the chat or in the comment section. Clear to all? And last, we are talking about these chromosomes. So how does a typical chromosome look like, by the way? So a typical chromosome would look like this, right? This is how it will look like. This is the P arm, this is the Q arm, and this one is the centromere or the primary constriction, right? And around this centromere, guys, around this centromere, there is this disc-shaped structure which is known as kinetochore. This is known as kinetochore. Now there are two discs. So these are two in number, two disc-shaped structures surrounding or around the centromere. Why are they present? Why are they present? So this is where the spindle fibers will attach, like this. So this is the site for attachment of spindle fibers. Hashtag site for attachment of spindle fibers. Anyone with any doubt, please let me know. So this was the basic structure of a chromosome. Anyone, guys? Multinucleated is primary endospermic cell. Yes, Pradeepa. Sorry, aaj late ho gaya. Why, Thakur? Anyways, and the second example, can you give me one more example of a multinucleated cell? It's your skeletal muscle fiber. Yes, syncytium. So yes. So this is about chromosome. Now chromosome, can be further classified into four categories. Depending upon the position of, depending upon the position of centromere, we have four types of chromosomes. Let me draw. So let's say if this is the centromere and the two arms are equal. So centromere is right in the center, that is one. Then it is slightly away. One is slightly longer, one is slightly shorter. And then one is significantly longer and one is significantly shorter. And last, it is almost at the tip like this. So we'll talk about these four different forms. First, this is called metacentric. This one is known as metacentric chromosome. This one is called submetacentric chromosome. This one is called acrocentric, and this one is telocentric. Right? So telocentric chromosomes, acrocentric chromosomes, you can easily get question on this topic in the match the following format or statement based questions, and it is already there as a PYQ. So in metacentric, centromere in the center, centromere in center, so both the arms, equal arms are equal, right? Submetacentric, slightly away from center. I am only talking about what? Centromere, right? Slightly away from center. So that means one arm is slightly longer, and the other arm is slightly shorter. While in acrocentric, it is quite away. It is centromere quite away from center. So therefore, one arm is quite shorter, other arm is quite significantly longer. And here, telocentric, centromere towards the tip. TT. Is that clear? So that's how we classify the two chromosomes. So we classify the chromosomes into 5 categories. Is that clear? Is that clear to all? Yes, please. Right? So one is metacentric, other is submetacentric, then we have telocentric, and fourth we have acrocentric. So these are the 4 different categories of chromosomes. Yes guys, please let me know. Okay Pradeep, I think I was confusing you with my other student Pradeepa. Sorry. Anyways, are these points clear to everyone? Done? Yes, please. So now guys, we are left with the one part of this, which is called, which is cytoplasm, right? Now I do have class at 7 p.m. by the way. All those students who have already joined my batches, I have class at 7 p.m., 7 to 9. We'll be starting with the next chapter. And there are amazing news coming ahead, but don't forget that that very soon, that is on 30th of August guys, 30th of August, I'm reminding you again and again, do not miss these tests or you will regret in 2027. I'm sorry to say, but yes, you need a special preparation. So first CBT is going to be conducted on 30th of August. So quickly get yourself upgraded if you're already a part of Unacademy's paid batches. All right guys, so we'll stop here. I'll take your next class day after tomorrow, Wednesday, and we'll be completing the chapter. All right, so don't forget about 30th of August, your first test, and this chapter is also included. So what are the rest of the chapters? Quickly find out. So subscribe and I'll see you soon. Bye.